Our next presenter is Dr. Mandy Barron, a senior researcher in the Wildlife Ecology and Management team. She's been working with us for the last 12 and a half years and specialises in ecological modelling of pest populations and diseases or interventions to control or eradicate them. Pre-COVID, Mandy enjoyed a 3-year retirement living off-grid on a tropical island in Tonga. And today she will discuss predator control findings of rabbits from 2 ecological restoration areas in the Hawke's Bay, City and Poutiri Ao, Tāne. So I'd like to welcome Mandy.
Hello. Yeah, I'd just like to probably get my slides up first. I'd just like to acknowledge my co-authors, Grant Norbury, also at Manaki Whenua, and Natalie de Burgh from Hawke's Bay Regional Council, who sort of instigated this project and provided the data. So what's the problem? Well, there's heaps, and we've heard a little bit about them today, there's heaps and heaps of predator-free programs being rolled out across the country. Big, large-scale programs aimed at controlling some predators. And there is a concern with landowners, understandably, oh, well, if we remove predators, won't we get truckloads of rabbits in response to that removal of predators? So this quote here is from a report done around the Cape to City predator control program, and yeah, basically saying this is a risk to the program, that a risk to landowner support and participation in such programs. So what do we know about what does control rabbit populations? So if we we can kind of split what factors affect rabbit populations into bottom up and so bottom up is resource limitations, things like food, burrow sites, and they're quite heavily influenced by land cover, what kind of vegetation, what the soil structure is there. But the food resources can also be impacted by competing herbivores. Yeah. And then above, at a higher trophic level, we have what we call top-down factors, top-down processes, and that's things like predators, stoats, ferrets, cats, hawks, people, and diseases, RH, rabbit hemorrhagic disease, and say coccidiosis, natural enemies, basically. Oh, sorry. That's right. I'm supposed to look at that. And I've put, oh, I didn't do that, did I? And I've put climate here sort of outside of all that, because climate can mediate both top-down and bottom-up processes, sometimes both at once, in indirect ways. For example, you might have climate affecting disease prevalence, or you might have climate affecting a high rainfall event which drowns the kits in the burrows. Or climate can affect how much food is available, flushes of green food that supports the rabbit population. So there's I guess the thing to take out of this diagram is there's a lot going on there. It's not just about predators. So what is the evidence that predators do regulate rabbit populations? And there is a review done by Grant Norbury and Chris Jones in 2014 that looked at exactly that. And what they sort of conclude is they did a review of different studies that had been done in New Zealand, Australia, and a couple from the Iberian Peninsula where rabbits are native to. And what they concluded was that in some areas where rabbit recruitment is limited by suboptimal environmental conditions, then yes, predators can regulate rabbit numbers. But in these sort of chronic rabbity areas, which show these eruptions, they have high rates of recruitment. And so in those cases, predators are ineffective at regulating rabbit numbers. And in fact, the only New Zealand study that had a non-treatment comparison looking at rabbit populations in the presence and absence of predators, and had some replication as well, was by Benjamin Bunny Reddix, and he looked at rabbit populations in North Canterbury and also in South Otago populations. And this was at the time that RHD had just come into the country. And the treatment was predator removal and non-predator removal at both sites. And what he found that was at his North Canterbury field site, so lowland, bit higher rainfall sites, there was a big difference between the predator and non-predator treatments. So in both cases, rabbit numbers were reduced with the RHD coming through, with the disease coming through. But, sorry, I lost my train of thought there. But there were bigger declines in the areas that still had the predators present. Then it's at the same sort of setup looking in the central Otago sites. They both declined, but there was no difference between the areas where there were predators present and not predators present. So that's probably the best study we have under New Zealand conditions, with, as I say, some non-treatment comparisons available. So amid these concerns and uncertainties, Hawke's Bay Regional Council contacted us to have a look at some of their study data, some spotlight data that they had on rabbit populations. So they do these spotlight counts routinely once a year or once every 2 years. And they just so happened that they had some that were in or out of predator control sites. So one of the, keep thinking I've got a pointer, but I don't, sorry. So one of the sites up here, up in the northwest area, Poutiri Ao, Tāne, and then another large scale predator control ecological restoration sort of area called Cape City, further down by the Cape. And they also, for the Cape City, they also had camera trap data as well. I'll just, then the predator control or the ecological restoration program in general started at different times. So Poutiri Ao was around 2011, and Cape City sort of cranked up 2015. So at Poutiri Ao, there were 2 sites with ongoing predator control. That was begun in 2012. And I've forgotten the names of those sites. Opawahi and Rangiora Trust that had the predator control, and then Woodstock, Tarawera and Riverland stations without. And they had spotlight counts so the routes were about 20 k's each in each of those sites. And they were done 2012, 2013, so annually at first, and then every couple of years, 2017, 2019, 2021. Cape City, that we had spotlight data available at 3 of the predator treatment sites there, and control there was rolled out in 2016, 2017. And there were 2 similar non-treatment sites, and these counts were done annually, so 2016 to 21. And as I said, the Cape to City also had some camera trap monitoring sites. Note that these were in different areas to the spotlight routes, so quite widely dispersed. About 30 cameras in each treatment and non-treatment, and they were spaced widely apart. I think it was 2 Ks in between, so they could be considered independent. The one advantage of this data set was that it had a year of pre-predator control monitoring as well. So you could kind of take into account maybe pre-existing differences between the sites. And the images, they weren't 100% sure of that they could distinguish between rabbits and hares in the photos that the cameras took. So they were kind of just lumped into lagomorphs, but probably okay for our purposes. So these are the results of the Poutiri Ao sites. The dots are the individual sites. We've got rabbits per kilometre and year along the bottom. And the blue arrow is when control first started. So you can see that all the sites started off pretty similar in terms of rabbit abundance, and they remain similar for the first 5 years of monitoring, although there was increasing variability there. Then a couple of years later, we get this huge spike in numbers in the rabbit numbers in the predator control sites. And the counts here, thing to note, the counts here were more variable between years than between sites, and also between the effective predator control. But that's what I just said. But statistically, because of that, no difference for the first 5 years, there was no difference between the predator control and no predator control sites, despite that large spike in the last couple of years. So these are the same sort of results, but for the Cape to City sites. And here it's different. The no predator control sites, we've got more rabbits in the no predator control sites, and very few rabbits in the predator control sites. The non-treatment areas remain consistently higher than the treatment areas, and there was no spike, say 5 years down the track after predator control started, like we saw on the other side. And again, here the counts were more variable between years than between the sites, but the effect of predator control was actually double that of the year and site effects combined. So yeah, a different story. And then we've got our camera trap data, which kind of tells a different story. Well, a different story again. And this, I tried to analyze it as a Backy design, which is before, after, meaning before and after control was applied. Control impact. So control here means the treatment control, and the impact is the treatment. And the treatment is judged to have an impact if the interaction between time period and treatment is statistically significant, because it indicates the response to treatment occurred after it was applied and wasn't just due to some pre-existing difference between the sites or some trend that affects all the sites. The other thing to note is that there's highly over dispersed, so most of the cameras were getting zeros, and then occasionally you'll get 50, 100 photos from one trap. And so you get these big outliers here. But you can sort of see that they started out pretty much the same 2015 and ended the same in 2020. 2016 is probably the biggest difference between the two treatments. But actually 2016 is really when the predator control, just like it was a month after the predator control started. So maybe that should have been in the before anyway. But in any case, there was no interaction effect. So there's no impact of predator control on the number of lagomorphs detected with camera traps. So we've found these conflicting results. What is controlling the rabbit numbers? And it doesn't appear to be predators, or at least it's not consistent from what we can tell with the spotlight and camera trap data. So yeah, this is just to show again them both on the same graph what was happening. Blue is the predator control areas, and in the solid lines, which is Poutiri Ao sites, predator control areas ended up with more rabbits. But then the opposite occurred in the Cape to City sites. So the blue lines, the blue dash lines, so then the next thought, well, maybe it's some kind of climate forcing. Maybe rabbit numbers are responding to some, rainfall events or just good years and bad years in terms of rainfall and vegetation type thing. But again, there didn't seem to be any clear signal in the data. Like it wasn't consistent. The changes weren't consistent between the sites. So there didn't seem to be some overall climate forcing going on. And having a model with a trend for year didn't really fit any better than having a model without it, or having maybe just a global yearly effect, but allowing it to be a wiggly, like a GAM, so a wiggly effective year. Wasn't a good model either. Having a site-specific year effect as a random effect in the model, that was the best fitting model. And that showed that there was more variation between the years than between the sites. So basically it was just showing that it was very site-specific what was going on, but we don't know where that variation came from. That's just the same graph again, but just showing that, well, there doesn't look to be that much synchrony going on there. Although I realised I'd cocked up when I was going over these slides that I've joined the lines between some of the missing years. So like 2016 was missing for the Poutiri Ao sites, and so I've just connected the line, whereas there should be a gap. Maybe they dipped down in between those years, or maybe they went up and came down again. Don't really know. So it's a bit hard to assess the synchrony there. So yeah, as I was saying, well, it looks quite site specific what's going on. What factors could be site specific? Hunting, poisoning, they're kind of site specific and year specific. We tried to get maybe some indication of what control had been going on there. Can't really get that for like, just someone going out and doing a bit of spotlight shooting or whatever. There was one large scale control recorded at, I think it was Puahi site, where they did fumigation and poisoning in 2012. But that was just one site, so it's a bit unbalanced to detect a difference there. The other thing, well, maybe it was disease, so RHDV, that can be highly site specific and year specific. It kind of, you get a wave one year and then you and then rabbit numbers crash. So there's few rabbits, so you don't get disease the following year, and then it might come back the next year or maybe 2 years later. So thinking, well, maybe disease could explain these fluctuations. So I tried to have a look at that, and there was the council do collect serology data, which is where they have shot samples of rabbits. They take a blood sample, and and then they see if there's antibodies to RHDV. And so that data was available, but it was only available for the non-treatment site. Well, there was a little bit available for the treatment sites, but it was highly unbalanced. So we only had that for the non-treatment sites. So it's kind of confounded what was going on there. So that wasn't, I mean, that still could be something, but I couldn't test it with the data we looked at. And it's, maybe in all probability, it's not just one factor. There could be indirect effects, things like climate, so high rainfall years and high rabbit density, you get outbreaks of coccidiosis. So there's like lots of multiple arrows going on there in our diagram. Or maybe it's climate, you get a high rainfall year, you get a bigger flush of vegetation, you get more breeding and recruitment, but also you might get density, you might get too many rabbits, so the rabbit's own density will affect what's going on, and they might show reduced survival and because of high rabbit density. Or there's, as I talked about before, there could be an interaction between RHD, rabbit density, but also that could be impacted by predators, as Ben's study showed, and also that's also influenced by resources, what the land cover is doing. Yeah. So back to my question, what is controlling rabbit numbers? We don't actually know. I can't actually say what the main factor was. It didn't seem to be predation. And these complex, identifying these key factors or identifying these interactions are hard to tease out of spotlight count data when you don't have the covariate data also at the same time in the same area. And quite also the predator effect, it was coarse because it was like, was there predator control in the area or wasn't there? And it would have been better to have an index of predator abundance to see if that was related rather than just yes or no predator control going on. The other thing is you need annual data to be able to check what the population dynamics are. Is there density dependence? Do they sort of peak and trough type thing? Or maybe if there is predator-prey interactions going on, if there's cycles, you need a long time series to detect that kind of thing going on. And yeah, I think because it was so variable between site and year, I think site-specific is a clue, and possibly it's a disease, it's possibly RHDV, a disease dense site regulation. But the good news was that predator control didn't seem to have a discernible effect on rabbit numbers. Overall, there was nothing in it. There were sort of two conflicting results. And so maybe that's enough to satisfy landowners that, we can't detect a difference. So don't worry about it. I don't know. And I guess in terms of, I was lamenting the lack of data in that as well, but maybe the monitoring data is enough just to show general trends in rabbit abundance over time. Are they going sky high or not? I mean, and are they going, are they getting really high once we start predator control? Maybe that's good enough. Note the opposite is not true. Rabbits control predator populations. when you have lots of rabbits, you have lots of predators. Thank you very much.